Bismillahirrahmanirrahim. Assalamu alaikum students. We continue our discussion on the topic of clinical significance of bacterial structure. Student, it means that why we study in detail the structure of bacteria. What is the importance of this studying of bacterial structure? How this study helps us in the field of medicine. Students, first of all, the most important characteristic feature is peptidoglycan. As you know, peptidoglycan is present in thick layer, multiple layers in gram-positive bacteria. Students, it is present in very small amount in thin layers in gram-negative bacteria. What is the clinical significance? The peptidoglycan thick layers makes the bacteria gram positive. That is, bacteria retain crystal violet color and they appear violet in color under the microscope. Secondly, students, peptidoglycan is only present in the bacteria. It is not present in the viruses, in human cells, in fungi, and etc. Number third, peptidoglycan is a very fine target for antibiotics. As it is only present in the bacteria, so it is a very fine target for antibacterial antibiotics. And lastly, students, the antibiotics which are the peptidoglycans, they inhibit the antibiotic, they attract the antibiotics are cephalosporins, penicillins, they target the peptidoglycans, vancomycin. Now we have got lysozymes. Students, lysozyme is present in human tears, saliva and other mucus secretions. And this lysozyme enzyme is antibacterial in character. That is, it inhibits the bacterial growth. Next, students, we have got lipopolysaccharides. Students, this lipopolysaccharide is present in the cell wall of gram-negative bacteria in the form of endotoxins. And it is because of this endotoxins that infections with gram-negative bacteria, they produce fever, decreased blood pressure and shock-like clinical signs and symptoms. Students, lipopolysaccharide also locates O antigen, and it is the presence of O antigen which is used to distinguish many gram-negative bacteria, for example E. coli bacteria. Students, next we have got teichoic acids. As you know, teichoic acids are present only in the gram-positive bacteria. They are absent in gram-negative bacteria. What is the clinical significance? Students, teichoic acids, they induce inflammation and shock-like symptoms produced by gram-positive bacteria. Now, we have got periplasmic space. Students, periplasmic space, it contains the enzyme beta-lactamases, and this enzyme, it inhibits the beta-lactam ring of antibiotics. So beta-lactamase enzyme, it inactivates the different antibiotics containing beta-lactam ring. Now we have got cytoplasmic membrane. Students, cytoplasmic membrane of a special species of bacteria, for example Mycoplasma bacteria, it contains sterols, and it is the presence of these sterols in the cytoplasmic membrane that these Mycoplasma are called as AFB positive bacteria, acid fast bacillus bacteria. Now students, we have got another structure, that is plasmids. Students, plasmids are very important and gaining more importance in today's field of bioengineering and biotechnology. Students, plasmids, they are extra chromosomal molecules of DNA. That is, they are located outside the DNA, outside the chromosomes. Students, these plasmids, they contain genes for antibiotic resistance. Number two, they carry genes for exotoxins. For example, exotoxins, which are enterotoxins released by certain bacteria like E. coli bacteria, anthrax bacteria, and even the tetanus toxin produced by Clostridium tetanus bacteria. These genes are located on plasmids. Next, students, we have got that plasmids carry the genes for bacteriocins. What are bacteriocins? These bacteriocins are toxic proteins which are produced by certain bacteria, and they are lethal for other bacteria. Now, students, we have got another structure important in bacterial structure, that is capsule. Student, capsule is the outermost structure of the bacteria. It is located outside the cell wall. Student, capsule is used as an antigen in preparation of different vaccine, which are called as capsular polysaccharide vaccines. Students, this capsule, it protects the bacteria from the phagocytosis of neutrophils and macrophages. Capsular polysaccharides are used in the identification of bacteria in laboratory, for example, Quell-Lung reaction test. Now, students, we have got flagella. Flagella are thread-like structures which helps in the mobility of bacteria. Students, two important bacteria, for example, E. coli and Proteus bacteria, they move from the urethra upwards toward into the urinary bladder, and they cause urinary tract infection. Flagella, they help in the attachment of bacteria to the human surfaces. And lastly, we have got pili or fimbriae. These thread-like structures, they also help in the attachment of bacteria towards the human tissue surfaces. So, students, these are important bacterial structures which are clinically important for our importance to study in